Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway. I'm sorry for the abrupt ending to the previous video. So we're discussing receptor tyrosine kinases at the moment, and we're specifically discussing this ErbB family of receptor tyrosine kinases, which contains 4 members here. Okay, so let's have a look at the structure of all ErbB family receptor tyrosine kinases. Okay, so firstly let me just generalize the structure of all receptor tyrosine kinases, and then we'll look at the ErbB family of receptor tyrosine kinases. So all receptor tyrosine kinases pretty much have a structure that looks like so. Okay, this has a few exceptions, such as the insulin receptor, which has a slightly different structure, and also what other receptors that are related to the insulin receptor, such as the insulin-like growth factor 1 receptor. Okay, but all the others, excluding those, look like so. They are a single polypeptide. Okay, so they'll have their amino terminus extracellularly. So these 2 parallel lines here represent the 2 layers of phospholipids of the cell membrane. So this is our cell membrane here, and we'll assume that this aspect above here is the extracellular fluid side of the cell membrane, and this side below is the cytoplasmic side. Okay, so the amino terminus is always extracellularly. Then what they have is a ligand binding domain extracellularly. Okay, so I'll colour this portion in orange here. So this is the ligand binding domain. Now the ligand binding domain is usually abbreviated to the LBD. So that stands for ligand, that's the L, binding, that's the B, and then domain, that's the D. Okay, right, so that's the main domain that you have extracellularly. Now, the ligand binding domain is obviously going to vary between the different receptor tyrosine kinases, okay, because they all bind to different ligands. So they'll all need to have different structure ligand binding domains. We will see the ligand binding domain specifically for our ErbB family receptors in a moment. Okay, then you have a membrane spanning alpha helix, which I'll show here. Okay, so this is a alpha helical structure which straddles the membrane and will have a large number of hydrophobic residues within it, which can interact nicely with the hydrophobic core of the phospholipid bilayer here. Okay, so this is an alpha helix. Then on the cytoplasmic side you have a tyrosine kinase domain. Okay, here. Now, this can either be whole or it can be split. Now, what do I mean by that? Well, basically, usually the portion which is actually going to make this tyrosine kinase domain will be all together in the primary structure, i.e., if you think about the polypeptide going linear like this, you'll have the tyrosine kinase domain all made up by one continuous region of the primary structure. Okay, so that would be a whole tyrosine kinase domain. However, some receptor tyrosine kinases, and it's not actually that uncommon, a lot of receptor tyrosine kinases do not have the two. Well, they don't have their tyrosine kinase domain produced by one continuous portion of the primary structure of the polypeptide. Instead, they'll have the 2 portions of the primary structure that are going to make the tyrosine kinase domain split like so. Okay, and then when it folds up into a 3-dimensional structure, these 2 halves of the split tyrosine kinase domain will be brought together. Okay, so in the 3-dimensional structure they'll be together, but in the primary structure they're not. Okay, so some receptor tyrosine kinases have a split tyrosine kinase domain like this, where there is this insert in the primary structure between the 2 halves that are going to make the tyrosine kinase domain in the tertiary structure. Okay, right, and that insert that you have between the 2 halves of the split tyrosine kinase domain, this is known as the kinase insert. Okay, right. So all of these receptor tyrosine kinases have this basic structure. Some of them will have a whole tyrosine kinase domain where there is one continuous region of the primary structure that produces the tyrosine kinase domain. And some will have a split tyrosine kinase domain where there are two regions in the primary structure that are separated by a kinase insert, which when the protein folds up, all these two split regions will come together and form the full tyrosine kinase domain. Okay, right. We'll move on to the ErbB family of receptor tyrosine kinases. I will come back to what the tyrosine kinase domain actually catalyzes in case you're not sure. It phosphorylates tyrosine residues basically, so this is an enzymic domain which has the potential to phosphorylate tyrosine residues on proteins. We'll come back to that later and I'll draw out pictures to show it. Okay, for now, let's look specifically at the ErbB family receptor tyrosine kinases. Now, I've said that there are 4 different receptor tyrosine kinases in this family called ErbB1, ErbB2, ErbB3, and ErbB4. Okay, these now also have other names. So firstly, ErbB1 is the most famous. Okay, ErbB1's other name is epidermal growth factor receptor, okay, or EGFR. So I think I should write this out in 4 at least once. So EGF stands for the epidermal growth factor, which is the ligand for all of these ErbB family receptor tyrosine kinases. Okay, so EGF is the ligand for all of them. Epidermal growth factor and then receptor. Okay, so ErbB1 is the most famous. It is the epidermal growth factor receptor. Okay, the others are then all related to EGFR, basically. Okay, so the other naming system for all 4 of them, so that's a special name for ErbB1, but there is a concurrent naming system that's also used. So ErbB1 is also called HER1, so it's got this 3rd name, and then ErbB2 is also called HER2, ErbB3 is also called HER3, and ErbB4 is also called HER4. Okay, now what does HER stand for? HER stands for human, that's the H, and then E stands for epidermal growth factor, so EGF, and then the R then stands for receptor. So that name makes a lot of sense, basically. Okay, so there are these 2 naming systems for the ErbB family receptor tyrosine kinases. You can either name them according to the ErbB naming system, or you can name them according to the HER naming system. And you then have to remember that ErbB1 is the most famous one. This also has the name epidermal growth factor receptor. Okay, right. HER2 is also very famous because it's the one that is famously overexpressed in certain forms of breast cancer. And the drug trastuzumab, the brand name for which is Herceptin, is a monoclonal antibody that binds to HER2 receptors and hopefully stops them from being able to be stimulated by epidermal growth factor, and is, I think, currently used for breast cancer. I think it's licensed and is used. Okay, so this is also known as Herceptin. Right, okay, so let's now draw a picture of what the ErbB family receptor tyrosine kinases all look like. So once again, they're made up from a single polypeptide. They have their amino terminus extracellularly, so I'll draw the cell membrane here. Now, we're going to study the ligand binding domain in a bit more detail because all of these ErbB receptors, they all have the same ligand. Their ligand is epidermal growth factor. So they all have a conserved ligand binding domain. So they all are very similar in structure. Okay, so we're now going to discuss the ligand binding domain of specifically this family of receptor tyrosine kinases. So this doesn't apply for the other receptor tyrosine kinases. Okay, this picture was the more general picture where we just drew a general ligand binding domain there in orange. Okay, so we're now going to look at this domain in a bit more detail for our ErbB family receptor tyrosine kinases. Okay, so basically all of them have 4 conserved regions, okay, which I'll show here, 1, 2, 3, 4. like so. Then they have the membrane-spanning alpha helix, and I'm now not going to bother drawing it as a spring-like structure. People usually don't, they just draw it as a straight line passing. But remember that that will always be an alpha helix. Whenever you've got whenever you've got a polypeptide going through the membrane, it's always an alpha helix. Okay, right, and then the tyrosine kinase domain, the portion with the enzymic activity, is going to be whole in the case of the ErbB family receptor tyrosine kinases, so they don't have a split tyrosine kinase domain. Okay, so here's our tyrosine kinase domain. Now let's study these 4 domains here. Okay, so basically you have 2 regions which I'm now colouring in green, which are the 2 leucine-rich regions. Okay, so these are regions which have a large number of leucine residues within them. Okay, so they are leucine-rich. So I'll just remind you of the structure of the amino acid leucine. Okay, so I'll draw the leucine amino acid as a residue. Okay, so when I say I'm going to draw it as a residue, what that means is I'm going to draw it as though it's within a polypeptide rather than as though it's a free amino acid. So here's the amino group here, and I will have the amino group as though it's bound to the carboxylic acid group of the amino acid in front of it. Then we've got the alpha carbon with a hydrogen coming off, and then the carboxylic acid group here. So that's the core amino acid structure now. The R group then of leucine consists of a methylene group, like so, then with a carbon coming off, and this carbon then has 2 methyl groups coming off, 1, 2, and then it also has a hydrogen coming off here. So this is the structure of a leucine residue. So basically, in these leucine-rich regions that you have within your ligand binding domain of your ErbB family receptor tyrosine kinase, you're going to have a large number of these leucine-rich regions. Sorry, you're going to have a large number of these leucine residues. So in these 2 regions, you've got lots and lots of leucine residues. Then, these other 2 domains that are in between, okay, so which I'm now colouring in yellow. Okay, these are going to be, again, rich in a certain amino acid, but the amino acid that they're going to be rich in is going to be different. So these are going to be rich in cysteine amino acids. So these 2 yellow domains are cysteine-rich regions. So, let me now show you the structure of the amino acid cysteine. So once again, I'll show it as a residue. So here's the amino group here. Then we've got the alpha carbon here with a hydrogen coming off it, and then the carboxylic acid group down here. Okay, the R group then of this cysteine residue is going to consist of a methylene group, like so, and then it's got a phyl group coming off. So this sulfur atom with a hydrogen attached to it, this is known as a phyl group. Okay, and it's got very similar chemical properties to alcohol groups because sulfur is in the same group of the periodic table as oxygen. Now, if you were to replace that sulfur atom there with an oxygen atom, it would be an alcohol group. Okay, as I said, sulfur is in the same group of the periodic table as oxygen. It's in group 6. Okay, so sulfur has very similar chemical properties to oxygen, and therefore we would expect this phyl group to have very similar chemical properties to alcohol groups, and indeed it does. Okay, so this is a cysteine amino acid, and in these cysteine-rich regions, we're going to have a large number of these cysteine residues. So all of these ErbB family receptor tyrosine kinases have ligand binding domains which contain alternating leucine-rich and then cysteine-rich regions, basically. Okay, right. Now, the ligand for all of these ErbB family receptor tyrosine kinases is going to be epidermal growth factors. So now let's talk about the ligands. Okay, right. Now, we want to talk about the activation then by the ligand of these ErbB family receptor tyrosine kinases. I'm now going to demote the ErbB family receptor tyrosine kinases to being represented back by this picture because it will save me drawing time to not have to draw this alternating leucine-rich and cysteine-rich regions. Okay, but remember the ligand binding domains will look like this. Okay, so what's now going to happen then is we are going to stimulate our cell with epidermal growth factor, okay, which is a peptide ligand. So what's going to happen is the epidermal growth factor is going to bind to the ligand binding domains of all 4 of these types of ErbB receptor. Okay, so here is the epidermal growth factor, let's say, in turquoise here. So this is our epidermal growth factor, or our ligand. Okay, and it is binding to the ligand binding domain of our receptor tyrosine kinase. Now, if you like, you can just imagine that this is the epidermal growth factor receptor, i.e. it's ErbB1, the archetypal one. Okay, then we've got our membrane-spanning alpha helix, and then our tyrosine kinase domain intracellularly down here. Now, what happens when the ligand binds to the ligand binding domain is that the receptor tyrosine kinase undergoes a conformational change, okay, which is specifically in this ligand binding domain region, okay, and this now means that this ligand binding domain can dimerize with another ligand binding domain on another receptor tyrosine kinase. Now, this dimerization has to be between 2 receptor tyrosine kinases that are in the same family. Okay, so you remember I told you that there are 58 different receptor tyrosine kinases, and there are 20 families of these. If we are talking about an ErbB family member that has just been activated by its EGFR, it can only now dimerize with another ErbB family receptor tyrosine kinase, because only the other ErbB family receptor tyrosine kinases will have similar enough ligand binding domains that they can dimerize with each other. Okay, so it can dimerize with any one of the other 3 epidermal growth factor receptors, or ErbB receptors. Okay, right. Of course, it can dimerize with an identical one. Okay, so if we imagine that this is the epidermal growth factor receptor, i.e. it's ErbB1, then it could dimerize with an exact replica of itself, so another epidermal growth factor receptor, but it's not essential. Okay, it can form these dimers where it has a different type of ErbB family receptor tyrosine kinase. Okay, so when you dimerize 2 receptor tyrosine kinases together which are absolutely identical, okay, so in this case we've dimerized 2 epidermal growth factor receptors together, or 2 ErbB1 receptors together, that's called a homodimer. Okay, so homo means the same, dimer means 2-membered structure, so this is a 2-membered structure where the 2 members are the same. Okay, if on the other hand we hadn't used an identical receptor tyrosine kinase, let's say in this slot we instead used ErbB2 or HER2, okay, to dimerize with EGFR, that would be a heterodimer. Okay, hetero means different, dimer again means 2-membered subunit, 2-membered structure. Okay, so this overall means a 2-membered structure where the 2 members are different. Okay, so but you can't go wild with the heterodimers. It must be another member of the same family of receptor tyrosine kinases. You can't, for instance, dimerize an epidermal growth factor receptor with a vascular endothelial growth factor receptor. Okay, so here are the 2 ligand binding domains dimerized together here, and they've both got this ligand bound to them, which is epidermal growth factor. Now, to keep this simple, let's imagine that it is an epidermal growth factor receptor that we've put in here, and we've got a nice homodimer. Okay, because that's simpler to picture. So here we go. Here is the tyrosine kinase domain of this second epidermal growth factor receptor here, which I'll colour in blue. So the idea now is that we have brought these 2 tyrosine kinase domains very close together. Okay, so we're now going to explore what tyrosine kinase domains actually do. Okay, so let's discuss what tyrosine kinase enzymes are. So not all tyrosine kinase enzymes are within receptors like this. There are 3 tyrosine kinase enzymes that are just within the cytoplasm. Okay, and what all tyrosine kinase enzymes do is they phosphorylate tyrosine residues. So let me draw you out a tyrosine residue. Okay, so once again I'm drawing a residue rather than the free amino acid. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, and once again I'll draw the carboxylic acid group as though it's bound to the amino group of the next amino acid along. Now the R group of the tyrosine residue consists of a methylene group, like so, with then a benzene ring coming off that methylene group, here, and then an alcohol group coming off the fourth carbon of the benzene ring. So this is a tyrosine amino acid residue. Okay, so this is what's going to be phosphorylated by tyrosine kinase enzymes. So we're going to stick a phosphate group onto this tyrosine residue. So let me show you the structure of a phosphate group. So a phosphate group consists of a phosphorus atom double bound to an oxygen atom. Then you have two alcohol groups coming off the phosphorus atom, one off either side, and then also a single bond from this phosphorus atom to an oxygen atom, which has then acquired another electron via ionic means. Remember, oxygen atoms need to gain two outer shell electrons to fill their outer shell. Okay, they'll gain one from this covalent bond here, so they have to gain another one, and that comes from ionic means, which means that they then get a negative charge. So this is the structure of a pure phosphate group, or an inorganic phosphate group. Okay, now in reality, tyrosine kinase enzymes do not take inorganic phosphate groups from the cytoplasm and stick them onto tyrosine residues. In reality, they take them off ATP molecules and then stick it onto a tyrosine residue. Okay, but to keep it simple, this will demonstrate the principle of adding a phosphate group onto a tyrosine residue. Okay, right. So how can this occur then? Well, if I circle this group here, which consists of the phosphorus atom double bound to an oxygen atom with an alcohol group, what does that look like? Okay, well that looks very similar to a carboxylic acid group. In fact, if I replace that phosphorus atom with a carbon atom, that will be a carboxylic acid group. Okay, ignore the fact that it's got 5 bonds coming off it in total, which is absurd for carbon, but just look at the portion that I've highlighted in turquoise. That looks very much so like a carboxylic acid group. Okay, and in fact it can react with alcohol groups in a similar way to how carboxylic acid groups would react with alcohol groups. So carboxylic acid groups would react with alcohol groups through esterification reactions, and we're going to perform something very similar in the case of this carboxylic acid-like group that we've got within the phosphate group. Okay, so we're going to remove that alcohol group from the phosphate group, like so. We're going to remove the hydrogen from the oxygen of the alcohol group. We're going to combine those two things together, okay, to make water, which is why this is a condensation reaction. And we're then going to bind the oxygen atom of the alcohol group that comes off the benzene ring of the tyrosine residue to the phosphorus atom of the phosphate group. Okay, and that link that you've created there is called a phosphoester link to show its parallels with ester links. Okay, so this is a phosphoester link. Right, so that's the principle of what a tyrosine kinase enzyme does. It phosphorylates tyrosine residues within proteins. Okay, right, so initially, these tyrosine kinase domains that we have on the intracellular aspect of our receptor tyrosine kinases, initially they're extremely inactive. Okay, they're not completely dead. They are capable of phosphorylating tyrosine residues on proteins, but they are extremely slow about doing it. They're not particularly active. Okay, so what's going to now happen when you bring these two receptor tyrosine kinases together like so? Okay, you have now brought these tyrosine kinase domains extremely close together. Now at the moment their activity is still ridiculously low. Okay, so they're not active really that much at all. However, they do have some activity. Now, each of these tyrosine kinase domains here is going to have a phosphorylation site, which is a tyrosine residue. So each one of these is actually going to have a tyrosine residue, like so. Okay, so the single letter code for tyrosine is Y. So when people put a Y like that, it means a tyrosine residue. Okay, so each of them has this special phosphorylation site, which is a tyrosine residue which can be phosphorylated. Okay, and if this tyrosine residue gets phosphorylated, this tyrosine kinase domain will become much more active. Okay, so it'll wake the tyrosine kinase domain up, basically. Now, what's going to happen is the tyrosine kinase domain on this first receptor tyrosine kinase is going to phosphorylate the tyrosine on the tyrosine kinase domain of the second receptor tyrosine kinase. So it will help just to label up these receptor tyrosine kinases here. So let's label up this first one as number 1 and the second one as number 2. Okay, so what's going to happen is, even at this extremely low level of activity, this tyrosine kinase domain of number 1 is going to end up phosphorylating the phosphorylation site on tyrosine kinase number 2. Okay, so it's going to be responsible for doing this, basically. Okay, now, when you stick that tyrosine residue onto the tyrosine kinase, that will activate it hugely. Okay, it then reciprocates and phosphorylates the tyrosine residue on the other one. Okay, now clearly it doesn't matter whether it was the first one that first phosphorylated the second one, or whether it was the second one that first phosphorylated the first one. The key understanding is that one of them is firstly going to phosphorylate the tyrosine residue on the other. That will activate this second one, and it will then go about more quickly phosphorylating the tyrosine residue on the other. Okay, so this process is known as transauto phosphorylation. Okay, or just as autophosphorylation. So some people will call this in full transauto phosphorylation. Some people will just call it autophosphorylation. So what does this mean? Well, we'll start with what autophosphorylation means. So auto means self. Autonomy means self-governing, so autophosphorylation means self-phosphorylation. So why is it called that? Well, basically you view this dimer of receptor tyrosine kinases as being one structure now, okay? And the tyrosines within this structure are becoming phosphorylated. Now, how are they becoming phosphorylated? Well, it's domains within the receptor tyrosine kinase dimer here that are phosphorylating the tyrosines within itself, basically. So this is self-phosphorylation if you view the dimer as being one structure in its own right. Now, the trans then kind of goes back on that by accepting that, if we view the two different receptor tyrosine kinases as being separate, what's actually happening is one is phosphorylating the other, and the other is phosphorylating that other one. Okay, so trans means across from. Okay, so, or on the other side. So, basically, the receptor tyrosine kinase across from it is going to be phosphorylated by it. Okay, so each one is going to be phosphorylated by the other receptor tyrosine kinase, so that's the trans portion here. Okay, now first we get the phosphorylation of these tyrosines within the tyrosine kinase domains itself. Okay, and this causes the activation of these tyrosine kinase domains, so their activity has gone up hugely now. Now they're not going to stop there. Now that they've been activated, they are going to phosphorylate loads of tyrosines that are in the rest of the cytoplasmic tail of the receptor tyrosine kinase. So for instance, all over the place in the cytoplasmic tail of the receptor tyrosine kinase, you have tyrosine residues that can be phosphorylated. I'm just showing 2 here, but in reality there will be many more than that. Okay, so there are loads of tyrosines that have the capacity to be phosphorylated by tyrosine kinase enzymes. Okay, these 2 are now active. Okay, so this tyrosine kinase on receptor tyrosine kinase number 1 is now going to phosphorylate the tyrosines in the cytoplasmic tail of receptor tyrosine kinase number 2. Okay, so it remains the case that the tyrosine kinase on number 1 is phosphorylating the tyrosines on number 2. And then, of course, the tyrosine kinase domain on number 2 is going to reciprocate and phosphorylate the tyrosines in the cytoplasmic tail of receptor tyrosine kinase number 1 here. Okay, like so. So you're now going to get lots of these phosphorylated tyrosine residues on the cytoplasmic tail of your receptor tyrosine kinase dimer. Okay, and these phosphorylated tyrosine residues are then called phosphotyrosine residues. Okay, so you're getting the appearance of lots of these phosphotyrosine residues. Now, it is these phosphotyrosine residues that now serve to recruit the effectors of the downstream pathways of receptor tyrosine kinase dimers. Okay, so many different proteins can come and bind to these phosphotyrosine residues. Okay, so there are special domains that you find in proteins which are capable of binding to phosphotyrosine residues. Okay, so just to illustrate the principle, let me draw one here. So, if your protein has a special domain in it that can bind to phosphotyrosine residues, okay, then you can come and bind to these phosphotyrosine residues on the intracellular aspect of the receptor tyrosine kinase dimer here, and that can do one of two things. Firstly, it can recruit you to a different cellular location. Okay, so if you were previously in the cytoplasm floating around, now you are going to be attached to the underside of the phospholipid bilayer. So that's one of the things that this can serve to do, recruitment. Okay, and secondly, the binding to the phosphotyrosine residue can also cause conformational changes, which can obviously affect your activity as well. So not only do we change your cellular localization, but we can also change your activity as well. Okay, now, two conserved domains that you find in a huge number of different proteins which are capable of binding to phosphotyrosine residues are SH2 domains and also PTB domains. Okay, and we're running out of space here. So let me go over the page and talk about SH2 and PTB domains. Okay, so we'll start with SH2 domains. These are more common by far than PTB domains. SH2 stands for Sark homology 2 domain. Okay, so the S is for Sark, the H is for homology, and the 2 is for 2. Okay, so these are domains that you find in a huge number of different proteins, and it's just a conserved structure that you can find in many different proteins which will bind to phosphotyrosine residues, at least generally. Okay, so it was originally found in this protein called Sark, which is actually a cytoplasmic tyrosine kinase enzyme. Okay, right. Then the other type of domain that's capable of binding to phosphotyrosine residues is a PTB domain, and this one's name is a little bit more sensible. It's just standing for phosphotyrosine, that's the PT, and then the B is for binding, so phosphotyrosine binding domain. Okay, so you can find PTB domains, which is again a different structure that you find conserved in many different proteins, which is then capable of binding to phosphotyrosine residues on the intracellular aspects of these activated receptor tyrosine kinase dimers. Okay, right, so we'll call it there for this video, and in the next video what we'll move on to is PI3 kinase. Okay, so we'll start looking at one of the downstream targets that these activated receptor tyrosine kinase dimers are going to recruit. Okay, so I want to stress they recruit a huge number of different downstream effectors and set off a huge number of different pathways. We're going to look at one of these downstream effectors, which is going to be PI3 kinase.